Hello, my name is Marina Konopleva. I'm a professor in the Department of Leukemia at MD Anderson Cancer Center. I will introduce today the topic of biology of BPDCN, understanding of CD123. BPDCN is a highly aggressive hematologic cancer which has poor outcomes with median overall survival only of 8 to 14 months. It is characterized by overexpression of the marker CD123 and some additional markers such as CD4, CD56, and some other ones. It is mainly represented in the bone marrow and skin, but can also involve lymph nodes and viscera. Right now, there are no approved therapies or standard of care, and the stem cell transplant is promising for selected patients in remission, mainly of a younger age. CD123 is the IL-3 receptor alpha. It is the transmembrane alpha chain of the IL-3 receptor, which is expressed at very high levels on plasmacytoid dendritic cells and also on the basophils. It is expressed at the low levels on some other hematopoietic cell types such as monocytes, eosinophils, myeloid dendritic cells, and also at the low levels on some normal hematopoietic progenitor cells. CD123 or IL-3 receptor alpha binds IL-3 and heterodimerizes with common beta subunit of GM-CSF IL-5 and IL-3 receptor complex, so-called receptor beta, to induce proliferative and anti-apoptotic signaling. CD123 is overexpressed in BPDCN, AML, and AML leukemia stem cells. On the low left, you see the RNA sequencing data of the CD123 expression comparing different subtypes of AML and normal progenitor myeloid cells. You can appreciate that CD123 is highly overexpressed in AML compared to the normal stem progenitor cells. Similar findings were found by flow cytometry, and the expression is the highest in the dendritic cells. In this case, these are the normal cells. On the right, the table represents the percent of CD123 positivity in different leukemia subtypes, including AML, MDS, also B-ALL, Hodgkin lymphoma, hairy cell leukemia, and additional lymphoid malignancies. IL-3, which is the ligand for CD123, is a cytokine which is required for in vitro generation of plasmacytoid dendritic cells. As I mentioned before, IL-3, IL-5, and GM-CSF receptors share the common signaling subunit beta. Each of these receptors is a heterodimer and is required to heterodimerize for the signaling. CD123 is IL-3 receptor alpha subchain. It is found, as I mentioned, on pluripotent progenitor cells, and upon binding of the ligand, it induces tyrosine phosphorylation, promotes proliferation and differentiation within hematopoietic cell lines. CD123 is very highly expressed on BPDCN cells. In this publication, the CD123 expression was compared amongst different BPDCN primary samples, and all of them have shown high levels of CD123 expression. On the low right, you can see the data presented at the poster at ASH 2016, which compared the levels of CD123 in AML and BPDCN. Even though AML overexpressed CD123, the expression was at least 10-fold higher in BPDCN primary samples. Due to high overexpression of CD123, it becomes a target for different therapeutic intervention. Here, there are multiple modalities which are now utilized for targeting CD123, and those highlighted in green have proceeded to clinical trials specifically in BPDCN. Amongst those is human IL-3 diphtheria toxin fusion protein, or DT-IL3, antibody drug conjugates, and chimeric antigen receptor T cells, or CAR T cells. The SL41 or IL-3 DT is a targeted therapy that is directed towards the IL-3 receptor alpha or CD123. It is a fusion protein of truncated diphtheria toxin fused to IL-3, which is a ligand for CD123. It binds to CD123 on the cell surface of the cancer cells, after which it internalizes. The diphtheria toxin is released and selectively kills the BPDCN cells that overexpress CD123. On the low left, you can see the activity of the SL41 in vitro against primary BPDCN cells, where the IC50 is in the femtomolar range. SL41 was shown to be potent against BPDCN cells in vitro and also in vivo in xenograft models. In the previous phase 1 study and a pilot study of a single cycle of SL41 conducted by Art Frankel, the major responses were seen in 78% of patients, 7 out of the 9 patients enrolled, with 5 complete remission and 2 partial responses. The subsequent trial led to the FDA approval of this novel compound, which is now known as Allogeneic. Additional modality to target CD123 is a novel anti-CD123 antibody conjugated to toxin. One of the agents is IMGN632, which is a product of ImmunoGen. It is a novel humanized anti-CD123 antibody, which is potent ADC conjugate. It has a novel DNA alkylating payload known as DGN549C. It is quite potent in vitro and in vivo model, and it is cleaved intracellularly, is more stable, and is quite effective in the preclinical models. This slide represents the preliminary data presented at the poster last year at ASH, which showed activity of IMGN632 in the primary models of BPDCN derived from patient relapse with BPDCN. As you can appreciate in the left lower corner, both the lower and the higher doses of IMGN632 completely reduce the tumor burden measured by the human CD45, CD123 positive cells. This led to the significant extension of survival and cures at the highest dose of IMGN632. Based in part on this data, the clinical trial with IMGN632 is currently ongoing and accruing patients with both AML and BPDCN. In the data presented at last ASH, the major responses were presented in 2 patients of BPDCN, which were primary refractory and failed to other therapies. In this slide, you can see the PET images of the 69-year-old female with BPDCN who was unable to complete SL41 due to liver toxicity, failed to standard chemotherapy, and was enrolled in this clinical trial. She had diffuse bone involvement, numerous lymph nodes and subcutaneous lesions, and 9 skin tumors. The PET images before and after cycle 1 show dramatic response to the IMGN632, a single cycle of treatment. And the skin lesions likewise have disappeared, as you can see on the lower right corner. Additional modality to target CD123 is CAR T cells. In particular, the allogeneic CAR T cells pioneered by the selectors was presented as a preliminary data at ASH 2016. These allogeneic CAR T cells are generated from the healthy donor and are genetically engineered to remove TCR alpha, and then they are transfected with CD123 CAR. They're subsequently being infused into the patient. Again, the preliminary data demonstrate that the primary BPDCN cells are highly sensitive to the CD123 CAR T cells in vitro in the left lower corner, and the treatment of the primary PDX with BPDCN demonstrated significant extension of survival and significant proportion of mice cured from the disease at the highest dose of UCART123. Again, this modality went into clinical trial, which is currently accruing patients. In summary, BPDCN is a highly aggressive hematologic cancer with poor outcomes, which universally overexpresses IL-3 receptor alpha CD123. CD123 is a membrane biomarker and a therapeutic target. Targeting CD123 is being explored by different modalities such as IL-3 diphtheria toxin fusion protein, antibody drug conjugates, and CAR T cells. Thank you for listening to my presentation today.